in the series of webinar. So Department of Environmental Science is organizing this is the second seminar webinar. So for this, I have a pleasant duty of welcoming the chief guest for this today's webinar, our Honorable Vice Chancellor, who is always encouraging us to do these academic activities. So for the benefit of the audience, and also I, on behalf of the department, I welcome our Honorable Vice Chancellor and also give brief introduction about our Vice Chancellor. So for the benefit of our participants, I would like to give brief introduction about our Vice Chancellor, Professor G. Hemant Kumar, who has done his MSc, BSc, MSc and PhD from our University of Mysore. And he has published more than 448 research papers in national and international journals and more than 150 research papers in national and international conferences. He has over 1,446 scientific citation and having the H index of 24 for his research contributions as of now. He has given invited and plenary lectures in a large number of national and international meetings and guided successfully to the 20 PhD candidates, and currently 8 students are working under his supervision. And he has sanctioned major research projects by DST, AICTE, UGC and DIETY, and also international collaborative research projects to the tune of more than 5 crores. He has been a member of science societies like Indian Society for Technical Education, life member of Computer Society of India, life member of Indian Science Congress Association, etc. He has conducted several workshops, national and international conferences in view of his academic excellence. Professor G. Hemant Kumar was awarded the Millennium Plaque Honor Prime Minister Award for contribution in the field of science and technology by Indian Science Congress Association in the year 2018, and Upadhyaya Samman State Level Award in 2017 from Upadhyaya Memorable Art Foundation, Udupi, and Teachers Day Award in 2017 for the Best Teacher from Lions Rotary Club of Mysore. He has been very successfully in designing and running the joint MOU programs between Unga University, Chennai and University of Mysore, and generating funds to the tune of 10 crores as academic collaborations with European Commission of Education and Network Research Project, where 6 premier institutes from India and abroad work together for the modernizing and enhancing Indian e-learning educational strategies. Earlier to becoming Vice Chancellor of Professor G. Hemant Kumar was served as different positions at the University of Mysore. He has served as the chairman of the Department of Computer Science for 9 years. He was Chief Coordinator Administration of Vignan Bhavan from the year 2009. A multidisciplinary project of rupees 100 crores from MHRD and 50 crores from UGC for supporting advanced research. And a coordinator Information Communication Division, coordinator Chinese Program, and director International Center, and coordinator e-governance and coordinator for implementation of high-performance computing system. Professor G. Hemant Kumar was the member of Maharaja Education Trust, Mysore, which is a premier education institution, governing council member of Gopal Swami Shishu Vihar Trust, Mysore, governing council member of Sharada Vilas Education Society, Mysore, member of Academic Council, Yurajas College, Mysore, Autonomous JSS College, Boys College, Mandya. Professor G. Hemant Kumar has visited several countries as a part of academic and exchange and research collaboration on invitation to Italy, Sudan, Moscow, Russia, Unga University, China, Wuhan University, China, Oman, UAE, Geneva University, Italy, etc. So on behalf of the Department of Environmental Science and on my behalf, I welcome you for this webinar. And also today we have another chief guest for today's webinar, Professor R. Shivappa, the President Registrar. So he is he was working as a professor and chairman before assuming his office as a registrar, and he has studied. He has a fellowship from Government of Denmark in 2000, and he has done master degree from the Bangalore University, secured first division, and he has many credentials for his teaching and learning with postgraduate students and guiding students of social work towards doctoral degree program on a regular basis, seeking assignment in projects management, research and relationship management for rural urban development programs, HRD and provisional personal management with the leading organization in India and overseas, and PhD with his rich experience in teaching learning projects, management, research initiatives, team management, personal management relationship, management in careers spanning 29 years in the bilateral NGO, academic and cooperative sectors. His expertise in steering projects on rural development, particularly in rural water supply and sanitation. He has significant experience in program design, planning, implementation, and introduction of innovative approaches in making the program owned by the people, and has knowledge of development and application of participatory strategies like PRA, participatory rural appraisal, PLA, participatory learning and action, and PMW, participatory monitoring and V techniques, advocacy of our policy reforms at government and intermediary levels in rural and urban development programs. An effective communicator with strong relationship management skills and hands-on experience in networking with consultants, NGOs, government agencies, and safeguarding the interests of beneficiaries and stakeholders. So, sir, on behalf of our department and on my behalf, I welcome you. So today also, I welcome our today's speaker, Dr. Rajesh Kumar T. He's Associate Professor and Ramalingam Fellow from Department of Biochemistry, JSS Medical College, Mysore, for today's speaker. So I request our Honorable Vice Chancellor to honor the speaker of today's speaker. Please come. I request our Honorable Vice Chancellor to speak a few words in this occasion.
So, Eldrigu, Namaskara. I am very happy to be here for this webinar on SARS-CoV-2 infection and COVID-19. Could environment pollution or diet impact? So this is the topic that has been given. So Department of Environmental Science is doing a lot of webinars. I think this is second or third webinar. So here our guest speaker, Dr. Rajesh Kumar, and our program chairman and also moderator, Dr. Ines Raju, and Dr. S. Shrikant Swami, all other faculties, students and research scholars of this department, and also our chief guest, Professor R. Shivappa. Today we have a right topic has been chosen for COVID-19, the environment pollution, and COVID-19 and environment pollution, how it is related, and speaker is going to speak. COVID-19 is a pandemic caused by SARS-CoV-2, which has been presented unprecedented health crisis. So economic distress across the world since December 2019. There are over 45 million cases and 1 million deaths of record worldwide. The patients with COVID-19 exhibit high degree of heterogeneity and severity of the illness ranging from asymptomatic to mild severe illness. Also, there is considerable variation in the number of cases and severity of the diseases within or between the cities and countries. What factor alter the risk of the COVID infection and severity of the disease among the population is not clear. Emerging evidence from across the world suggests that ambient air pollution could be a significant dominant factor of the infection, transmission, and severity of the COVID-19. The present the presentation which will focus on pathobiology of COVID-19 and share the emerging data, how environmental pollution impacts on SARS-CoV-2 and infection and COVID-19 diseases. Lastly, the presentation will be discussing on
Department of Environmental Sciences.
Lastly, the presentation will discuss about photochemicals and also food plants which can alter the risk for the COVID-19. So understanding how, when, and what types of settings for the SARS-CoV-2 spreads between the people is critical to develop the effective public health and infection prevention and measures to break the chains of the transmission. So current evidence suggests that the transmission of the SARS-CoV-2 occurs primarily between the people through the direct, indirect, and the close contact with the infected people through series infected secretion, such as saliva and respiratory secretions. Though there is not the respiratory droplets which is expelled, and when it is infected, persons coughs or sneezes and talks and sings. So the high quality research is needed to elicit the relative importance between the different transmission routes and the role of airborne transmissions, and also this absence of the aerosol and also generating procedures, the dose of the virus required for the transmission is to occur, the setting of the risk factors for spreading the events, the extent of the asymptotic and the pre-asymptotic transmissions. So with this, I would say that today's webinar is very essential for the current situation. The Department of Environmental Science is a very dynamic department in our campus, working well with the different applications, because many of the subjects which will not be touching the society, these departments always touching the society, causing some good cause in the society. I congratulate all the faculty members for conducting this. I hope this webinar is going to give a good turn of what we call the environment and pollution, and also the COVID-19, how it can be related. Thank you very much for giving me an opportunity to this thing. I am very happy to inaugurate this seminar. Thank you.
So I request our registrar to speak a few words.
Yes. Good afternoon, everyone. More than 79 viewers, participants are taking part in this webinar. Our Honorable Vice Chancellor has set a very good precedent, very good foundation kind of a thing to understand this particular topic. In my view, I think we are fighting with this COVID-19, and a lot of challenges are being faced, and we need to understand how we can combat this pandemic. I think through this kind of webinars, understanding the nitty-gritties, understanding the challenges, and try to helping, we need to try and help one another to mitigate this pandemic situation. And multidisciplinary approaches will help us in understanding the pandemic and also to address the challenges. Definitely, this environmental aspects of understanding the problem is going to help us. I'm extremely happy that Environmental Science Department is conducting series of webinars, especially on COVID-19, and I'm happy that I'm participating here, and expert is there here, so he is going to speak on this particular topic. I wish all the best, and listeners, the viewers, kindly raise your hand and also ask all the questions after the end of the presentation. So once again, I wish all the best for this webinar series. Now over to Professor Shrikant Swami.
Introduce the speaker to the forum.
Good afternoon, everyone. Our Honorable Vice Chancellor is always supporting us in almost all the activities, and also our Registrar, Professor Shivappa, is also supporting us in almost all the academic activities. Thank you, sir. Thank you for inaugurating this webinar. And I'm immensely happy to introduce today's speaker, Dr. Rajesh Kumar, Associate Professor, Ramalingaswamy Fellow, Department of Biochemistry, JSS Medical College, JSS Higher Education, Mysore. Dr. Rajesh Kumar got his MSc degree from Bangalore University and later pursued his PhD at Central Food Technological Research Institute, Mysore. He was awarded PhD degree by University of Mysore in 2001. After his PhD, he moved to Johns Hopkins University, Baltimore, USA for his postdoctoral studies and continued to work as faculty at Johns Hopkins University for next 14 years. While in USA, he also founded a biotech company called as Curveda for discovering drugs for respiratory diseases. In 2015, he returned to India after receiving prestigious Ramalingaswamy Fellowship from Department of Biotechnology, Government of India. This program was initiated to bring Indian scientists working abroad back to the country. That is actually known as reverse brain drain to enhance the R&D in India. After returning from USA, Dr. Rajesh joined as associate professor at Department of Biochemistry, JSS Medical College, JSS Academy of Education and Research, Mysore. He is carrying out research to investigate how environmental air pollution causes respiratory and heart diseases. His team is also involved in discovering and developing herbal medicine and dietary supplements for treatment of these diseases. Along with the research which he is carrying out, he is also doing teaching. He teaches students who are pursuing MBBS, BDS, MSc, and BSc degree. Dr. Rajesh has around 60 research publications, 3 patents with H index of 32 and citation over 700. This is very remarkable. 7,000 citation for any researcher is one of the good achievements. Yes, published in impact journals such as Nature Medicine, Nature Medicine, Journal of Clinical Investigation, Science Translation Medicine. In last 5 years, he has received over 12 crore worth of projects from Government of India, from different agencies like DBT, DST, DBT, Welcome Trust. This is also a very important thing for any researchers to carry out his research. He is currently monitoring as guide and co-guide for 10 PhD students. He is a member of several academic societies and ad hoc reviewer for scientific journals. I'm really happy to introduce to the audience. Thank you. I request Dr. Rajesh Kumar to continue this work.
Thank you, sir, for introducing me. Thank you, the Honorable VC, Honorable Prime Minister, sir, for being here and inaugurating this conference. And I'm very thankful for Dr. Raju and Dr. Raman for inviting me here. When Raman asked me to present a seminar here, I was deliberating what topic should I choose, because I work on air pollution. And then I thought maybe COVID-19 is impacting the world and particularly India. So I thought maybe I can have some data showing how air pollution can impact COVID-19 and how it can help in public health intervention program. So if everything is okay, I can start. So maybe we can go to the next slide. I can use this one?
Yeah.
Okay.
I'll share.
So my topic is SARS-CoV-2 infection and COVID-19 disease, how impact of environment and diet influences the disease process. Before I start my lecture, I want to define few terminologies so that it will be easy for the audience to know what I'm talking. First one is COVID-19. COVID-19 refers to coronavirus disease 19. It's a disease. So it appeared in 2019, so it's given the numerical as 19. There's also terminology called SARS. SARS refers to severe acute respiratory syndrome. And again, it's a disease which appeared in 2002. CoV is coronavirus. SARS-CoV refers to the virus that caused SARS in 2002. And SARS-CoV-2 refers to the virus that is causing COVID-19. Okay, so it is also called as novel coronavirus. Just to differentiate between the SARS-CoV from the one which caused SARS, from the COVID-19, they have added this numerical as 2, so SARS-CoV-2, they are related, they are closely related to each other. And the other terminology is zoonotic disease, which you often hear in the news. What it means is this disease, the COVID disease or the virus, has jumped from animals to humans. So that's why we call it zoonotic disease. And we have encountered many zoonotic diseases, and COVID-19 is one. Previously, we have encountered AIDS virus. HIV is also a zoonotic disease. Ebola is a zoonotic disease, Nipah disease. Many are there which we are dealing with most often. Can I go to the next slide? Yeah. So this is the outline of my talk today. I'm going to start with the COVID-19 statistics and timeline, and then tell you what do we know about SARS-CoV-2 so far, whatever information we have, and how does SARS-CoV-2 infect and cause COVID-19 disease. This basic information is very important, mainly for the students as well as being academician. We should be able to be inform the common man about this SARS-CoV-2 and also clear some misinformation or misconcept about this disease. And my main focus would be to talk about how air pollution affects SARS-CoV-2 infection and the disease severity. Why is this important at this moment? It's important. Why? Because it's important to identify environmental factors or any modifying factors that can alter the severity of the disease, or it can protect from the disease. For example, if air pollution is involved, and if there is a lot of data involved, and there is a lot of data out there showing that air pollution can impact COVID-19, and because of that data, the government of India had banned cracker use during Diwali. So these kind of data helps the policymakers to come up with policies so that it can reduce the severity of the disease or even the transmission of the disease. Lastly, I'm going to talk about various dietary components that can affect the immunity against COVID-19. That will be a very small part. The main focus would be on the pathobiology and the air pollution effect. This is the statistics. As of today, there are almost 60 million people are affected with COVID-19. The death is around 1.4 million people. As you can look at from here at the left side of your the slide, these are the ranking. US is still leading. Next comes India, Brazil, France, Russia. So it's a serious disease. And in terms of deaths, US is still leading. China is still not in the top 10.
They have not disclosed that.
We can argue on that, but so number speaks about it. So the death-wise, India is ranking at number 3 position. And if you look at the map of COVID-19 cases in India, it is affecting every city, every village, mainly both infecting the metropolitan cities, the Tier 1, Tier 2 cities. So this is the timeline of COVID-19 pandemic. Early December of 2019, last year, or rather 2019, the first case was recorded, which was a pneumonia case. And the hospitals here in the Wuhan were unable to identify what is the cause for that pneumonia. By the time they realized, by end of that month, that is by 31st December, already 27 cases of pneumonia were reported with unknown cause. So this led to the panic. And the panic was because all these 27 cases were related to with one seafood market, which is Wuhan seafood market. They were either directly or indirectly related with the people, or they had visited to that seafood market. And the seafood market not only sells seafood, it also sells a lot of animal meat as well as live animals. In China, there is a culture where they like to have exotic animals, which could be, porcupines, snakes, crocodiles. That's the culture. It's not something offensive, but that's the culture because it's a privilege for them to have exotic animals. So this seafood have meat as well as live animals, and often people who are close, who go for buying and all this stuff, it is possible that the virus present in the animals could jump to humans. So that is what happened. The panic for this was that the same in 2002, similar incidents happened almost the same month. They had in 2002 similar case of pneumonia, and they were later found to be SARS. So because of similarity being that pneumonia, also it was from seafood, so they thought that this is something serious. So then they sent it to the virology lab, and in the lab they sequenced the virus, and they found that it's a novel coronavirus which was not seen before. That means it's a coronavirus, but it was a novel. It's a new one. For the first time, they found that. And later, the virus started spreading. And by January 13th, one case outside China border was identified. And by January 20, the transmission was going very rapidly. And they confirmed that there is a human-to-human transmission. By January 23rd, the Wuhan city was locked down. So then by January 29th, 34 provinces across China was reporting the cases of coronavirus. So fast, we can see it from December 8 to end of this January, you had already the whole country got infected. And by January 30th, WHO declared that it's a public health emergency of international concern. So alerted this time. In February, the WHO raised the risk level at the global level. And in early February 11th, the international the taxonomy organization of the virus, they named this novel virus as SARS-CoV-2 because the SARS-CoV-2 is related to the SARS-CoV which caused SARS in 2002. So to differentiate from the early one, they gave it the new name as SARS-CoV-2, and the disease they called as COVID-19. Coronavirus disease 19 because it appeared in 19, and rest is all history. We all know about the statistics. So this is the Wuhan city, the seafood, where you have all the meat. You can see all kind of animals are sold here. So let me tell you little bit about SARS-CoV-2. It's an enveloped virus. That means it is having a membrane. It is a positive sense single-stranded RNA. Single-stranded RNA act as a genome as well as mRNA. We all have DNA as a genome, but this virus has RNA as genome. The genomic size is around 30 kilobase pairs. The size is 80 to 120 nanometer. That means it's almost 100 nanometer, which is almost like 1/100th of the hair diameter of your hair. So that's small it is. So that's why it can reach very deep into your body. And if you look at the structure, it has on the surface, it has these projections called a spike protein. These are the spike proteins. And because of the spike proteins on the surface, it gives a corona effect. It gives a kind of halo effect. So that's why the name coronavirus. The whole family of viruses have this corona appearance. So it has a spike protein on the surface, and then below that is the membrane glycoprotein along with the envelope proteins. And below this membrane is your genome. The RNA genome is present, which is associated with nucleocapsid protein. So this is just a small amount of information to know to the students. And if you look at the family of Coronaviridae, there is a subfamily called Coronaviridae. And under that, there are so many genuses: alpha corona, beta coronavirus, gamma coronavirus, and delta coronavirus. So the SARS-CoV-2 belongs to beta coronavirus under lineage B. And the same SARS-CoV which caused SARS in 2002 also belongs to the same lineage. The another important coronavirus is the MERS coronavirus, which is the Middle Eastern Respiratory Syndrome that was the cause for outbreak or rather epidemic in Arab countries, in the Gulf countries. If you look at the similarities between these two viruses, it's 79%, and with the similarity between SARS-CoV and MRSA is 50%. So this tells you that how closely they're related. So they were all lineage from the same family. But with time, with evolution, they kept on accumulating more mutations, and now they become totally new ones. So the natural host for SARS-CoV-2 is bats. Bats, for some reason, is a good breeding animal for them. It does not kill the bat, it just grows there, it breeds there. And the intermediate host, during the evolution of these viruses, they have undergone some mutations, and that helped them to jump to another host. In case of SARS-CoV, which caused SARS in 2002, civet cat was found to be the intermediate host. That means it jumped from there to here, and it can live here in this animal also. In case of MERS-CoV, it is a camel. The camel was spreading to humans. In case of SARS-CoV-2, still it's not clear, but pangolins are one of the natural intermediate hosts. It does not kill pangolin, and pangolin is well consumed. It's one of the favorite dish in China. So that people suspect that probably the humans got infection from big contact with the pangolins. So the intermediate host has transferred this virus to humans, and now we know there is human-to-human transmission, either by contact transmission, which could be direct contact with the infectious person, like handshake, or you touch a contaminated surface, or it could be a droplet transmission. When we speak, when we sneeze, you can release respiratory droplets, and these respiratory droplets could have viral particles. And while speaking, the respiratory droplets can travel almost 6 feet from here. That's why social distancing is important. There is also data suggesting that it could be airborne. So what is the airborne? It doesn't mean that it travels in the air. It means that these droplets, the smaller droplets, can remain in the air for longer time, and it can travel for longer distance, beyond 6 feet. So there is data suggesting that it could be airborne transmission also. Now, if you want to compare the coronavirus outbreaks in the last 10 years or rather 20 years, you know about SARS-CoV-2. Excuse me. Yeah. So here, that started in 2019 and still continuing. SARS-CoV was first reported in China. It ended in 2004, started at 2002. The total number of cases are around 8,000, and the death was around 774 patients.
You could please continue. Sure.
So the death percentage is around 10%. In case of MERS, which was first reported in Saudi Arabia, and a few cases are still reported, it's continuing. The total number of cases during that period, 2012, was around 2,520, and the total number of deaths were 866. So the rate of death, the percent death, was 34%. That means this virus was much more deadly than what we are experiencing today, the SARS-CoV-2, which is the death is very, very less, 3%. Now, I'll talk a little bit about pathology of COVID-2. How does the virus cause disease? Now, if you look at this virus, I told you the virus is having these spike proteins. The spike proteins are the one which can latch to the receptor called ACE2 receptor, which is expressed by the lung cells. So when you inhale, the virus gets into your lungs, and the cells are having this receptor called ACE2, which is hanging outside, which is protruding out of the surface of the cell. And when the virus moves around, it can latch to this ACE2 protein. ACE2 protein is a receptor protein, but it's also enzyme, okay? So which is involved in the blood pressure control. There is one more protein, enzyme, which is also closely associated with ACE2, and that is a protease enzyme. That basically what it does, it chops off this head of this spike protein. When it chops off the head, you are unfolding and then exposing the fusion machinery. This is a kind of a protein which latches to the surface of the lung cell, and then it forms a opening, and it basically it fuses with the host cell membrane. And now the virus is able to introduce the RNA into the host cell. Remember that virus will not survive outside your body. It needs a host. Okay, so if you prevent the entry into the host, it will die within time. That's why social distancing is very important, so that you stop the transmission. Now, once the virus enters into the cell, it undergoes translation, and it produces certain proteins. One of them is the RNA-dependent RNA polymerase. This enzyme helps to make the many copies of this genome. At the same time, the translation will produce a lot of viral proteins. Now, the whole machinery of the cell, your cell, is being hijacked by the virus to make its RNA and proteins. So basically, it's exhausting all the machinery, all the energy in your cell so that the virus survives and makes more copies. Once the genome is made, many copies of that, the protein interacts with the genome, and you have a mature virion released out. When it releases out, the cell which has got infected is also killed because there's no more energy inside the cell to survive. So every time one virus infects, you have hundreds and thousands of viral particles generated, which is released into the tissue. So now, if you want to target or the SARS-CoV-2, or if you want to mitigate the disease, the best way is you can prevent the infection of the virus. How can you do that? Either you can mask the spike protein, because spike protein is the one which is latching to the cell receptor, ACE2, and it prevents it from entering into your cell. Or you block the ACE2 receptor. If you block the ACE2 receptor, the virus cannot bind to it, and therefore it cannot enter your cell. So that's one the drug targets. So vaccines, what have been generated now, produced are being targeted against the spike protein. So the antibodies will coat these spike proteins, mask it, and it will not allow it to bind to ACE2. That's one mechanism. The other cell mechanism is T cell mediated immunity, wherein the cells, the T cell will recognize the infected cell and kill it. So that's how the vaccine is being developed. But there are also the chemical drugs which are targeting the RNA-dependent polymerase, which basically, if you inhibit this enzyme, the genome will not be copied, and therefore you can halt the transmission. And there are different steps at which you can target with the drugs. So ACE2, remember this ACE2 receptor, it's very important. It is the one which helps to virus to enter your cells. So ACE2 is expressed across many cell types. In addition to lungs, your gut, heart, eyes, liver, bladder, kidney, pancreas, brain, prostate, placenta. So it tells you that the virus has potential to infect every cell in your body, most of the cells in your body. So wherever it goes, that tissue gets affected, and therefore you will have symptoms related to that organ. Okay, so you may find that, the virus is a respiratory virus. Why is it I'm getting a kidney failure, or why am I getting diarrhea? That also. Now, just to show you, this is the lung cast. This is left lung, right lung, and this is the trachea, and as the trachea goes down, it branched into two bronchus, left and right, and then the bronchus further is branched into primary, secondary, and tertiary branches. So at the end, you look at the surface. This is the buds. So I can show you more clear picture. So this is the trachea branches into bronchus, and finally the bronchus further divides, and finally you end up into these structures, this grape-like structures. They are called the alveolar sacs. Okay, and this is the scanning electron microscope picture. You can see it looks like a buds coming out, and if this is at higher magnification. Now these are where the air gas exchange happens. So if you take a cross section of this is how it looks. We have each of them is a sac where the air is trapped, and the blood vessel is there between this. There's a septa between two alveoli, and the blood vessel is there here, and the air circulation or the air exchange happens there. This is a cartoon showing that how each of these alveoli is well vascularized. It is supplied with the capillaries, right? So this is a cartoon showing that. So this is the alveoli, and this is the capillary close to it. So the gap between the alveoli and the capillaries is kept at thinnest possible. Why? Because the gas from the blood vessels has to move from the blood vessel into the air sac, which is the CO2, and from the air sac it has to enter to the blood vessels. So the oxygen goes from here to here, whereas carbon dioxide goes from here to here. So the gap is kept minimal. Okay, so this alveoli is lined with two types of main cells. One is called as type 1 alveolar cells. These are the lined ones. And these are the type 2 cells. So type 2 cells have two functions. These cells, they can produce type 1. So every 48 or 72 hours, you have repairing of the old cells, new cells are lined up. At the same time, this type 2 cells also produce surfactant. And surfactant, this yellow, whatever lining you see, that's a surfactant. So surfactant is very important to prevent lower the surface tension and prevent the collapse of the alveoli. If you don't have the surfactant, the end of each of these alveoli will collapse. So if you collapse, means there will be no air trapped here. So understand how the biology of your air exchange happens. Now, SARS infects. SARS can infect your nasal as you inhale it because it's airborne. Your nasal cells can infect it. It goes into the trachea, down to the primary, secondary, and finally reaches to the alveoli. It reaches to this part. When it reaches to the alveoli, it starts infecting this cell particularly. What is this cell? This is a type 2 cell. And the function of type 2 cell is to produce surfactant, right? As well as L1. If this dies, there will be no surfactant produced. So that means your lung function is declined. So that is what is happening here. So I'm just going to show you the cartoon here. So if this is the alveolar cell, just to give an example how this cell happens. If this is a cell, expanding it here. This is the cell, and this is the surrounding the cell is a space, and the blood vessel is the side. Okay. So the virus, by the help of ACE2 receptor, enters your cell, then undergoes replication. Also, at the same time, activates certain proteins, which are transcription factors called NFKB and LRP, which start secreting the cytokines called TNF alpha, IL-1, IL-6. The function of TNF alpha, IL-1, IL-6 is it helps two things. One is it recruits more immune cells from the blood into the lungs. So what does it mean? It means that this TNF, IL-1, IL-6 will make this capillary very porous. It's now permeable. So which helps the WBCs, which are in the blood vessels, to migrate into the lung space, into the alveolar spaces. So you see with time, you'll have more of these immune cells accumulating, and they start producing more of TNF alpha, IL-1, IL-6. So that terminology, what we have is, when you see this accumulation of these cytokines, we call this cytokine storm or cytokine release syndrome. Okay, this cytokine release syndrome does many things. It recruits more of these inflammatory cells. These inflammatory cells, they start secreting free radicals, proteases, and cause acute lung injury. Okay, to show you much more clearer, if this is your alveoli, this is the blood capillaries. Now, the cytokines and chemokines, they are making more permeable this capillary, which as a result, your WBCs will go inside. Along with your WBCs, even the plasma goes into your alveoli. That means your lung is now getting filled with the fluid. And that is what we call pneumonia. Okay, and when this whole, instead of air, now it's filled with water, basically the blood plasma. And now it is interfering with the gas exchange because the gap between the sac and the capillary is broadened. And thus also there's a liquid here. So CO2, which is in your blood, can easily cross this, but oxygen cannot enter. So when you have this, you have a pneumonia. And at the same time, because oxygen is not being crossing into the bloodstream, so the patient has what we call hypoxemia. So you have pneumonia, and because it's hypoxemia, your brain is stimulating your lungs to work more. You have shortness of breath, you have cough. So these patients, they have shortness of breath and cough. At the same time, cytokines which are released, they can go to the brain and increase temperature of your body. That's why you have fever. With the continuous progression of this inflammation, slowly what you see is the type 2 cells are dead. When type 2 cells are dead, there's no surfactant. Now the alveoli collapses. And the gap is filled. The gap between the alveoli and the capillary is filled with plasma, dead cells, immune cells, plasma proteins. Now this whole thing is called as hyaline membrane. Now see what you can see. The oxygen is not entering into it, and the carbon dioxide is also not going. So the blood oxygen level drops significantly. When the blood oxygen level drops significantly, you have a problem in your other organs to work. So this is a picture showing how a normal alveol looks and how a ARDS looks. Now, if you look at under the X-ray, if you take the X-ray of this patient, you see this whitish background here. That is all because of the fluid present here. The more darker it is, that more healthy it is, and the time it does. So this is what is happening in the lung. So now, It also, when because of cytokine storm, because there is high levels of the cytokines, they are released into the bloodstream. Now they go and spread across your body. And wherever they go, they cause these capillaries very porous. Now what happens when it becomes porous? The blood just leaks into the tissues. At the same time, because there's injury to endothelial cells, there is a thrombosis. That means there's a blood clots. So you have a leaky vessels, and leaking will cause blood volume go down because it's leaking everywhere, right? So the blood volume goes down. When the blood volume goes down, your blood pressure goes down. Okay, and it is now because it's leaky, the organs are not well perfused. That is, the blood is not flowing good, and so it's not well maintained. So even the organ is depleted of nutrition. So the organ damage starts happening. So you have a multi-organ failure. So now you see the patient with COVID-19, they show fever, they show shortness of breath, they have cough, they have what we call as multiple organ failure, you have ARDS symptoms. And because the cytokines can go anywhere into your system, it can go to brain, then it is showing a multiple organ effects, right? And that's why the patients show a very heterogeneous phenotype, symptoms. Some are asymptomatic, some are symptomatic. Among the symptoms also, you'll see that some patients complain that they are losing taste and smell. Could be that it's a direct effect where the virus is going into the brain and trying to do this, alter this function, or even the inflammation can cause all this process. Some people complain of diarrhea. The lung is a common organ, but people are dying even because of cardiac dysfunction. So it's a whole body which is getting inflamed and affected. So if you want to now block or prevent the infection, what you can do? I told you that you can block this ACE2. That means researchers have found that if you can knock out this receptor, the virus cannot enter the cells and it cannot make you sick. So that is the first target organ. If you have drugs that can block these transcription factors and reduce cytokine storm or cytokine response syndrome, the patient severity disease is less. That means patient recovers faster and goes home. So there is a drug called dexamethasone, which is now used in the clinic, which has been shown to be very effective in improving the patient health and reducing the mortality. So if you have a drug which can prevent the blood clogging or thrombosis, that is a good target for improving the health. So you can see multiple drugs are being used for the management of COVID-19. But there is not one drug which can effectively cure it. So you have to do multiple drugs. Ideally, you would like to prevent the entry of the virus itself. If you can block that, you can protect the patient or the subject from the disease. Now, air pollution and COVID-19, that's the topic of the day, right? So how is air pollution impacting COVID-19? I told you that COVID-19 is a highly heterogeneity. That means the symptoms are wide variety. Important things, 80% are asymptomatic. And who are symptomatic, they show from mild to moderate disease, and some show very severe pneumonia and ARDS. Why is that? Clinicians and researchers are very puzzled that why is that 80% people are asymptomatic? And it particularly affects older people and had minimal effects in children. And one data suggests that this minimal effect in children could be because of ACE2 expression is very less in children compared to the older people. Why is that some cities and countries and ethnic populations are more affected than the others? Right? Same virus infecting, showing different symptoms. Why is that? So now the question is, what factors are contributing to the heterogeneous susceptibility, transmission, severity, mortality rates within and between countries? So this could help in public health intervention. If you know this factor is contributing to higher mortality or higher transmission, the government can bring certain rules and prevent that happening, right? So that's a reason it's very important. Is it genetic? Maybe some race, some ethnicity population are more resistant to the virus, or is it diet? Because diet is varying from veg to non-veg to vegan to, so there is many things. And then in diet also you have high calorie, low calorie, rich in green, non-green, all that part. How about air pollution? Is air pollution different? Because across the globe you have different levels of air pollution. How about the population density? ethnicity. So there are so many factors we don't know what is contributing to the increased transmission, severity, and mortality within and outside the countries, right? So my question is long-term exposure to air pollution a modifying factor? What does that mean? Long-term means if you're living in an area with high levels of air pollution, are you having greater risk of developing COVID-19 disease? And this is not something a theoretical question, because there is a hard data showing a strong link between viral respiratory diseases and air pollution. So this is not something new. It's already there. If you take influenza, the flu virus, very well shown that more the air pollution in that area, more is the influenza infection and severity. And now there is new evidence suggests that populations being exposed, very important, being exposed to long-term air pollutions are more susceptible to COVID-19. Okay, I'm going to share the data with that. What are the sources and key air pollutants? As you all know, automobile emission, biomass cooking, burning of the waste, natural calamities like volcanoes, forest fires, crop firing, industrial emissions, and then also anthropogenic activities like burning crackers. What are the key air pollutants in our ambient atmosphere? PM 2.5, that's the particulate matter of size 2.5 microns. PM 10 of size 10 microns. NOx, SOx, ozone. WHO recommends these, according to recommendation, these are the permissible limits. The annual mean has to be around 10 microgram for PM 2.5. And for PM 10, it has to be 20 microgram per meter cube. NOx 40, SOx 20 microgram. But look at Indian permissible limits. Annual is 40, that means 4 times the WHO recommendations. 20, this is again 3 times more than the annual limits. So the permissible limits is very high for Indian context, but it is, compared to WHO ones. So now the question we are asking is whether populations being exposed to long-term air pollution are more susceptible to COVID-19. There are 3 hypotheses to which people are generated data. One hypothesis is that maybe the PM, the particulate matter, acts like a carrier, acts like a vehicle to carry the virus to a longer distance. Or maybe the second one is, could it increase its susceptibility and also the infectious rate? Third hypothesis, maybe it increases the risk of severity and death by exacerbating the immune response and organ injury. Maybe it's aggravating the ongoing injury to the lungs or the heart or to the blood vessels. That may be one reason. So all 3 are potential hypotheses. All 3 are very valid assumptions to be made. So let's go one by one. For the data to supporting the hypothesis 1 is that this group, Frontera from Italy, they compared the COVID cases with the NOx air pollutant. And they found that for some reason, this is where the NOx is very high, and this is Italy map. This Po Valley is in the northern area of the country, side of the country, and it is covered with, it is surrounded with these mountains. So the climatic condition is very different from the rest of the country. So what they did is they found that for some reason, the infection was very high, and also the death was very high. If you look at here. So this is the reported number of cases. You can look at the map. The darker, the more deaths were there in that area. The cases comparatively, relatively, deaths were also very high. Now, if we compare this map with NOx or PM 2.5, it matches very much. What does that tell you? Probably there is a role of air pollutant increasing COVID-19 cases, increasing the death in those positive cases. Same thing in China also. This is the PM2 concentration across the country. And this is the map of COVID-19 cases. You can see more air pollution there is more COVID-19 cases. So these authors thought maybe because here in this area, the climatic condition is very, very different. The air is very stagnant because it's surrounded with mountains. The air is very stagnant. There is a high humidity. it's not the wind flow is less there. Could be that PM is acting as a carrier. Okay, and this is their hypothesis. Maybe the viral particles, they are sticking to the PM, and therefore they are being held suspended in the air for longer time, and it can travel for longer distance. 6 feet is the one which we think while we're talking the droplets can fall apart. But now if it is in sticking to the PM, it can travel meters. It could be 50 meters. There are reports where in the New York, the whole building of apartments, there was a case at the bottom, because the vents are all linked to each apartment, the cases rampantly increased, but they were not knowing what would be the cause. It could be that it is traveling along with the dust particle to the other places also, other apartments also. So a valid point that the virus could be sticking to the surface of the PM and could be carrying away to the longer distance. And this is a paper published in New England Journal of Medicine where they compared the viability of this virus, both COVID-1 and COVID-2, on different surfaces, also on aerosols, like aerosols, which are, they are in the air. The virus can remain live even up to 3 hours. That means if I sneeze and if that has a virus in the droplets, in this room, it will remain for 3 hours in this room, active. So after me exiting this room, if somebody comes, even after 3 hours, there's a possibility that that person can get infected. That's something scary, right? And they also tested different surfaces. When the virus is kept on, if it is spotted on the copper surface, the virus can survive till 4 hours. In case of cardboard, again, till 8 hours it is survival. Stainless steel, till 24 hours. Plastic, almost 24 hours, it can remain. So these are the surfaces which we come across. If it is contaminated, you can suspect that that could be, if it is, the virus could remain. And all this test was done at a temperature of 20 to 20 degrees Celsius and at 40% humidity. So you can take this data, based on where you live. If the temperature, if it is inside your home, yes, this is very close to that. But if it is outside, like if it is 30, 35 degrees, probably the viability of the virus will change. Okay, so that's very important. So now you put this equation, that is PM is being sticking to the surface, and it is surviving for 3 hours. So based on the wind flow, these PM can move across many distance or many meters. So that was the reason the hypothesis could be true, that in this area, the Pope Valley, the infection, high infection could be because PM could be a carrier. Okay. Then coming to the hypothesis 2, can air pollutant increase the spread of COVID-19? I told you that COVID-19 the virus gets into your cell only using the ACE2 receptor, right? So if ACE2 expression is high, that means you have increased risk of getting infection, and there is increased risk of catching the virus also from other person. So this study, what they did is they compared the ambient air pollution, particulate matter, 2.5 and with the rapid spread of COVID-19 in US. And what they're looking was the basic reproductive ratio, which you call R0 value. It is basically defined as the average number of individuals that an infected individual could infect an entirely susceptible population. Basically, how many people would I infect if I am sick? So that is defined as R0 value. It is a dimensionless epidemiological parameter to estimate the spread of a disease through a population. Probably you could have heard in the news 6 months back, wherein the task force is always coming up with numbers. Oh, maybe it is infecting the ratio. The spread is very high. We have to do more, quarantine and social distancing measures. It's because you want to decrease the R0 value. Okay, so it's a very important parameter of public health officials and policymakers because it indicates the onset of an outbreak based on the threshold value of 1. If it is less than 1, that's a good news because it's not spreading that high. You're spreading to another person or maybe less than 1 person. So it'll die with time. But if it is more than 1, that means you're spreading, one guy is spreading to 3 people, 3 will make it to 9 people, and 9 people to 27 people. So it's exponential way it is spreading. So you have to be worried about that. So now the question was, is air pollution increasing or not value? So, so they did some modeling, used a modeling approach where they compared the state level PM 2.5 exposure with the R0 value of the state wise in US. And they also considered many potential confounding factors like gaseous air pollutants, meteorological variables, race, population density, age, hospital beds, ICU beds, median household size and income. Many cases, there are many factors they included in the data. And what they found is that this is the map of US. This is the PM 2.5 concentration. So you can see the R0 value increases wherever the air pollution was high. What does that tell you? It tells you that in states where there was higher air pollution, the R0 value was very high. That means one person was infecting more number of people than the normal case. What they concluded is with 1 microgram increase per meter cube was associated with increase of R0 value of 0.3 units. And this algorithm was following a trend until 6 microgram per meter cube. Beyond 6 microgram, the model fell apart. Now, this is 6 microgram per meter cube. Guess what? In our country, what is the levels of PM 2.5? The national permissible limits is 40. And today, across the country, in Delhi, it's 300, 400. In Northern Hemisphere, because of the winter conditions as well as crop burning, it goes beyond 200. So this is very high compared to what we have, right? So the bottom line is there is a good correlation between the level of PM and the R0 value. And they compared this pollution value from from 2012 to 2017. Okay. Now, how is it doing? This is another study where they took the lung cells and then exposed it to PM10. And their question was, is PM increasing ACE2 expression? I told you, if you have greater ACE2 expression, the entry of virus becomes easy. The more ACE2 expression, the easy of viral entry. And when they exposed it to the lung cells, they found that the expression of ACE2 on the surface of the cells was very high. That means it's doubling or 3 times high when you expose it to PM10. Even if you expose it to a cigarette smoke, if you're a smoker, your ACE2 expression will be very high. That means you are having a greater susceptibility to get virus infection. So it is possible that PM, long-term PM exposure, increased the ACE2 levels, and therefore the R0 value, which is the secondary spread, is more. Makes sense, right? So, and this is supported by previous data from influenza virus infection. There is a strong relationship between air pollution levels and influenza-like illness in Beijing. This was a study published where they found that they compared the incidence of influenza, the flu virus-like symptoms, from 2008 to 2014. And it barely appears during the winter period. I can see the temperature is down. But during the winter, whenever the pollution levels were very high, the incidence of influenza was also very high. Here, this is December. So it clearly tells you that whenever the pollution is high, temperature is low, the chances of getting sick is high. Then they compared the risk with every increase of 50 micrograms. With every increase of 50 micron per meter cube in the PM levels, the relative risk also increased. So clearly it tells you that high air pollution can impact the sickness in case of influenza, and they are finding the same kind of with COVID-19 too. Make sense? So that is hypothesis 2. Now, going to the last hypothesis that long-term air pollution exposure increases COVID-19 death. First one, increase in the transmission. Second one, increase in infection. Third one, is it increasing the death? All three are related to each other, right? So now, this is a study just published, I think last month, a couple of months back, where they compared the COVID deaths in US in around 3,000 counties and compared with PM 2.5 level, historical PM 2.5 levels from 2000 to 2016. So you're talking about almost 17 years of PM levels with COVID deaths. So it's a historical exposure. That means you are in an area where there's high pollution and they're looking at the death in that population. So look at this data. So beautiful it is, right? If you look at the PM levels, half of the country, the eastern part of the country, rather from center to eastern, is high polluted. And then this is the California belt. Here also you have a lot of industries, so this pollution is high. And if you now look at the depth in this area, exactly matches. Half of this country, this eastern part, east hemisphere, is having higher deaths. Again, in the California belt and Arizona belt is very high depth. So it clearly points out that in the areas of higher air pollution, the death is also high, related to COVID death. And the authors found that the percentage increase of COVID-19 mortality risk per microgram meter increase of exposure of PM 2.5 was 8%. That means for every microgram increase in the air pollution, it was 8% increase in the death. That's a big number. Again, these values are this is, you're talking about 20 microgram per meter cube across this country, right? We have 40 at the start, it goes till 300, 400. So now you can just imagine the impact of air pollution in our country related to COVID deaths. Unimaginable. Now, this is the data from Italy. This is Italy was severely or seriously affected in the early phases of COVID-19, and they also found the same thing. With the increase in the ambient PM, the number of ICU admissions also increased. Okay, so the conclusion of this study was infection is widespread in regions with more pollution. Patients in polluted areas presented with more severe forms of disease requiring ICU admissions, intensive care admissions. The mortality, the death was twofold higher in polluted regions than regions despite similar rates of ICU admissions. So what does tell you? That though the number of admissions in two areas were same, the area with higher pollution had twofold higher risk of getting admitted. The patients showed more. And the circle shows the proportion of population over 65 years of old. So this area had higher number of people with above 65 years old. So again, clearly showing there is a link between air pollution exposure and the ICU admissions. This is the data from China. What they did is they compared the time-dependent association of particulate matter pollution. That is, the previous studies were all long-term. This is, and that particular day, what was the level of air pollution and how did it impact the COVID-19 case fatality rate. Look at, this is from January 19 till March, and the dark green represents PM10 pollution and the light green represents PM2.5 pollution. Whenever the PM10 pollution increased or PM2.5 increased, after 2 days, you see an increase in mortality. There's a lag period. So 2 days before, when the pollution was very high, after 2 days, you see more mortality due to COVID-19. So it matches with the pollution increase, more mortality. This is a correlation coefficient plot showing that if you look at the p-value, it's so significant, clearly suggesting that air pollution can increase the risk of death because of COVID-19. This is another study from China. Again, the same thing. Regions where they had more of air pollution, the cases were also very high. Again, very good significant correlation between the pollution, PM2.5 levels, and the cases. This is the most recent one where this group calculated the percentage of mortality caused due to COVID-19 with air pollution as one of the factor. Okay, so you can see this map shows the estimated percentage of COVID-19 mortality attributed to air pollution from all the anthropogenic sources. China, you can see dark, that means more deaths were here, more percentage of deaths were here. US, in this area, the pollution is high, so the more deaths. In India, half of the country is dark, so we have more percentage death here. So what did they conclude? They came up with a number. This is a number. In Europe, 19% of the death that happened due to COVID was because of air pollution. East Asia, where India also belongs, 27% is attributed to, 27% of death, COVID death is attributed to air pollution levels. Okay. So now this is at the country level. In Czech Republic, 29% of mortality among the COVID-19 was related to COVID because of air pollution. That means you could have reduced 29% of death if the pollution was less. You can take the other, you can interpret in the other way also. Okay, so these are different countries' values. I think for us, what is interesting is India, 17%. That means 17% of COVID-19 deaths are attributed to air pollution. That means if you can reduce the air pollution level, these many people, almost 23,000 people could have been saved. That motivates us to reduce air pollution. That's what this data suggests. I think this number 17% is at lower end, because if you look at the air pollution, this is the pollution levels today's, okay, PM2.5 in major cities. You can look at here, at least in Bangalore and all that, we are safe, around 55, 60 is there. But look at the Northern Hemisphere, 200, 180, 180. Delhi is hidden somewhere in this map. It is about 200, about 300 today. So now if you can look at the map of COVID cases across India, you can see most of the metropolitan cities have higher numbers. So there is a correlation, though it's association studies, right? So in summary, the chronic exposure to PM can increase ACE2 expression, that can increase the infection rate and severity of the disease. More the ACE2 expression, more severe symptoms are there. Okay. At the same time, this PM can suppress your host defenses against the virus and aggravate the injury, because PM is also associated with adverse respiratory diseases like dry cough, conjunctivitis. So when you have these symptoms and you add up COVID-19 to that, it worsens it. So that means the injury is fastened or accelerated, and therefore these patients will have more severe disease. Okay. What is the limitation of this data? Whatever data I presented is all association studies, wherein they have taken the ecological variable. That means air pollution in that area correlated with number of COVID deaths. But if you really want to say air pollution is a causal factor for the death, we need to know the air pollution levels at the individual level. We need to know at that if that individual had a smoker, or was he having diabetes or hypertension, because we know that the COVID-19 deaths are more in those individuals. That data is not available. We are looking at area and comparing to the COVID deaths in that area. So that is the limitation of this data. But the models, very clearly suggest that there is a link between air pollution exposure and the COVID-19 deaths. Now, if you look at the, and most of the studies, what I presented was retrospective studies. From the past data, you take it, analyze it, and then project it. So we need to do a prospective studies to show whether in areas where there is more air pollution, whether the death was really high. But the data suggests it is, but it needs hard data to support it. Now, coming to another area, can diet influence the risk for COVID-19? Right? This is another modifiable factor. You all know, if you have these diseases, diabetes, hypertension, cardiovascular disease, or any kind of respiratory disease, you're likely to have severe outcomes from COVID-19. Again, obesity, people are more obese, they also show severe outcomes of COVID-19. And there are a number of factors that contribute to these conditions, but diet is another important factor. So people believe that diet could be an important modifier of health in these COVID-19 patients, but data is very scanty, so it's still not clear. But we come across every day about the news, about the articles showing that food items that can boost your immunity, right? If you look at the number of foods that can increase immunity by different mechanisms, it could be that it increases vitamin C, many other components are there. So we were also interested in last 3 or 4 months to identify identify plant-derived phytochemicals that can work, help in preventing COVID-19. And we searched and we identified, along with other many people, that these two could be potential phytochemicals. One is curcumin, which you all know is present in turmeric. The other one is quercetin. It's a flavonoid present in onion, tea, apples, many green vegetables. Sapsige soppu has a lot of this quercetin, right? And we made a case that these two components could be used as prophylactic or therapeutic agents and confer benefits for COVID-19 management. Why did we claim this? Because both the compounds show antiviral activity, broad spectrum antiviral. They are anti-inflammatory agent. They can suppress, calm down the immune system. They also have anticoagulant effects, okay? So this is the antiviral activity curcumin. You can see it can show antiviral activity against many viruses, including the SARS-CoV. Even quercetin has been found to have antiviral activity against coronavirus, respiratory virus like flu virus, as well as many other viruses. So, and FDA has categorized these two phytochemicals as generally recognized as safe. That means you can consume it safely. And there are clinical trials done where they have tested 12 grams per day of curcumin to be safe. For quercetin, they have gone till 5 grams per day. Okay, so even at highest level, you have minimal toxicity. So ideally, you would like to test this curcumin or quercetin whether it protects from COVID-19, right? But nobody has done that because it needs a lot of funding and money to do this. But another important reason why people have less faith in curcumin is that curcumin, though you eat, within half an hour, it is cleared from your body. So curcumin, which is present in turmeric, cannot remain in your body more than half an hour. So the efficacy is totally down. On the other hand, quercetin can remain in your body, can build up in your body, and it can reach to a concentration that can be clinically efficacious. Okay, so although if funding is there, we could do a trial, but we don't have trial. But in place of a clinical trial, what we did is we just to show that curcumin and quercetin can work. It can directly inhibit the virus. It can block ACE2. There's a data there out. Okay, the bioinformatic data was showing that curcumin can interact, quercetin can interact with ACE2 and block its viral entry. It can inhibit these transcription factors. Therefore, it can reduce the cytokine storm. Quercetin can also be antithrombotic. It can prevent blood clotting. So it has many effects. So primarily in mouse models, people have shown that curcumin and quercetin, if you supplement, it can protect from viral-induced acute lung injury, pneumonia, as well as ARDS. So with that background, it makes sense that these two components are potential phytochemicals which can work for that. But what we did was, we developed two products for COVID-19. We basically coated a face mask with curcumin or quercetin. The idea is that here people are not consuming, it's safe. If the virus comes in contact with the mask, it should immediately get killed. And with that idea, we have tested this mask against various bacteria, which are respiratory bacteria, which causes pneumonia. Also a simplex virus, herpes simplex virus. This virus also have an envelope, so it is almost kind of similar to the coronavirus. And we found that it is very effective in killing this virus. And we also made this moisturized gel. It's a non-alcohol. It's an alcohol-free gel or germicidal sanitizer, you can say. And we found that it is effective till 2 hours post-application. That means if you apply and till 2 hours, if the gel remains on your hand, it can kill the germs. Whereas if you take the commercial sanitizer, it does not work after 2 minutes. Why? Because the alcohol gets evaporated. So currently we are testing against SARS-CoV-2 too. We're testing at being sent to a lab in Johns Hopkins University, and we kept our fingers crossed to see if the results are positive. So this is something we did in the last 3 months. So in conclusion, long-term exposure to high levels of air pollutants is a significant cofactor that influences severity of COVID-19 outcomes in COVID-19 patients. Okay. Diet rich in anti-inflammatory phytochemicals such as curcumin and quercetin may help in reducing severity of COVID-19. And finally, still vaccine is not there, no drugs. So maintain social distance, use mask, sanitizer, and protect yourself and others from COVID-19. So I'm going to stop. That's my last slide. These are my team. I would like to acknowledge the grant support from DBT as well as DST. And I would take questions and answers.
Thank you very much, sir, for your excellent presentation. If you have any questions, if you have an audience from the questions, the speaker will answer for the questions.
Anybody can ask question.
Lydia, chart.
ಚಾಟ್ ಬಾಕ್ಸ್ ಅಡಗಿಲ್ಲ ಈಗ ಬರ್ತಾ ಇದೆ
ಈಗ ಬರ್ತಾ ಇದೆಯಾ?
Is there role in the PM
size?
COVID virus?
If there is one study where they've shown that PM 2.5 and PM 10
good correlation with COVID-19. The size, good impact, but PM10 is filtered in the upper respiratory tract, it does not go deep into your lungs. So though there is association, but the effect of PM 2.5, because of its size, it can reach to the alveoli, and the injury happens more in the alveoli for causing the ARDS and pneumonia. So PM 2.5 is more important than PM10.
Now, I have one question. What about the genetic factors?
Yeah.
Because the people throughout the world, the different races having different genetic factors. Is there any influence?
Yes, certain minority population like blacks, Latinos are more susceptible for COVID-19. What genetic factor is responsible? We don't know yet. So data has to be looked up for. Because this is the data coming up now, right? So most of the papers what we see are being derived from the data which is publicly available. Nobody has done a study to take the samples and do a genomic sequence to find out the data. But I feel definitely because 80% of people are asymptomatic. Why are they? And this asymptomatic means they don't have. It doesn't mean that they don't have virus. They are the same number of viral particles are there between asymptomatic and symptomatic. They are spreading the virus.
Then can we call them as carriers?
They are carriers. They spread it. So they are well shown.
So what about the herd immunity?
See, herd immunity as per the immunological definition, if more than 50% of your population is infected, the chances of transmission is less because it 1 to 1, it goes less to 1, right? The R0 value becomes less than 1. So that's good. But still, we don't know that herd immunity is kicked in India or anywhere else. So we don't know.
In USA,
Sunappa,
they are not taking any spicy items.
start ಆಯ್ತಾ
Maybe
ಮೀಟಿಂಗ್?
it's spreading of virus.
Huh?
So higher than India. And some telling that India using the spices and also they develop a lot of immunity compared to other.
Based on the data, like US is number 1 and India is number 2. So that argument that spices are protecting us may not hold. Hardly the number is not,
Second place.
Yeah, that is third. So we can't say that. Okay, but yes, I believe in that data, but not very.
About the mask.
Why did we use cotton mask for the purpose of yeah? So cotton mask is more technical aspect. The coating of cotton is easy than the non-woven mask. So and we have found that after coating even till 10 washes the mask was effective. And cotton is more user friendly, environmental friendly, and also skin friendly. It does not itch. People have allergy to non-woven mask. That's how it is.
Thank you, sir.
Yeah, with this, we can conclude.
Any questions?
What is the size of corona?
The size is around 100 nanometer, 80 to 120 nanometer, which is 1/100th of your hair diameter.
So yes, you said how do you block the rest of the?
One is the antibody. Either you block the spike protein, which is by antibodies. Vaccination is based on that. You are raising the antibodies against the spike protein, so it goes and mask it. Or you take a chemical that will block ACE2 and does not allow the virus to sit on the ACE2. That is two ways.
Any such chemicals?
There are, but these are all not specific. Like for example, I told you about the quercetin. Quercetin is according to, as per the computational modeling, they show that it interacts with ACE2, but there's no hard data.
So what is the breeding period?
Okay.
So if there is no questions, we came to the last part of the session. Good afternoon, one and all. Respected Vice Chancellor, Honorable Vice Chancellor, and also respected Registrar, respected Speaker, or dear Chairman, also our colleague Dr. Srikant sir. So today, first we went with this brochure. First Vice Chancellor. Vice Chancellor read this. Why can't you invite this professor to present? This is related to his current issue. Why can't you invite professor to our department, our university. We said, okay, sir, we'll invite. Then sir has accepted, and on behalf of sir is encouraging this, our honorable Vice Chancellor encouraging to do this type of work. So we requested, sir has accepted without in spite of his busy schedule, sir came and also inaugurated this. So on behalf of our department, on behalf of our University of Nature, we are thank you very much, sir, in this. And our registrar, sir. And also we went to register, sir also said, okay, I will come. You arrange this, we'll see there. Sir, on behalf of Department of Environmental Science, thank you very much, sir, for coming over here and also inaugurated this. And our speaker, sir, we thought, sir, I think maybe spending very less time to prepare this. Then we asked the sir, no, we want to start this, at least we should collect some information related to this to prepare the correct information, he said. And we, sir, has accepted this. And he prepared many slides related to this, how this COVID virus and also this air pollution interacts, and also related to this, how it is spreading, how this air pollution is directly or indirectly influencing, also causing these diseases. When the air pollution is high, maybe the transmission and also infection is high, and rate of death is also high. As he observed, and also he compared many slides, taking Indian and also other USA places. So on behalf of Department of Environmental Science, on behalf of our students, thank you very much, sir, for your presentation. Thank you, sir. Thank you. Okay.
Thank you very much,
Dear
sir,
student,
for nice
and,
presentation.
thanks for the arranging this in this university. Thank you very much. Thank you, one and all. Thank you, sir.